Taenia Solium is a species of tapeworm cystod transmitted to the human through the consumption of raw or undercooked infected pork. To understand the symptoms, diagnosis, and treatment of this worm, we must first understand the life cycle of the pork tapeworm in detail. The pork tapeworm consists of a large number of segments or proglottids. Because the tapeworm is hermaphroditic, each segment or proglottid contains both male and female reproductive organs, specifically an ovary and a testis. Eggs develop inside the gravid proglottids, and these eggs, along with the gravid segments, are then passed out of the infected human body with the feces. Pigs ingest contaminated food or feces containing the tapeworm eggs. The eggs of the pork tapeworm require gastric acid, digestive enzymes, and bile salts to hatch and release the larvae. After the eggs pass through the stomach and the small intestine of the pig, they hatch to release the larvae known as oncospheres. The larva or oncosphere penetrates the intestinal wall to enter the bloodstream and subsequently develops into larval cysts or cysticerci within the muscles of the infected pig. These larval cysts are transmitted to humans through the consumption of infected raw or undercooked pork. Once inside the human host, the larval cysts develop into adult tapeworms which attach to the wall of the small intestine using the suckers and hooks located on the worm's head known as the scolex. In the life cycle of the pork tapeworm, there are two hosts. Pigs serve as the intermediate host because the eggs hatch into larvae within the pig's digestive system. Humans represent the definitive host because the parasite matures from a larva into an adult worm inside the human body. However, humans can also act as an accidental intermediate host when they ingest food contaminated with human feces containing these eggs. Upon exposure to gastric acid and bile salts, these eggs hatch to release larvae or oncospheres capable of penetrating the human intestinal wall. They then migrate through the bloodstream to various bodily tissues where they develop into larval cysts or cysticerci. Perhaps the most critical of these tissues is the brain, leading to the formation of neurocysticercosis within brain tissue. The clinical presentation of Taenia Solium infection depends on the specific life stage ingested by the human host. For instance, consuming infected raw or undercooked pork results in intestinal taeniasis, which presents with symptoms such as abdominal pain, diarrhea, and nausea. Conversely, the ingestion of eggs is significantly more dangerous due to the development of cysticercosis. The most critical of these is neurocysticercosis, which presents as new-onset seizures in adults accompanied by headache and focal neurological symptoms. Diagnosis of a patient infected with the pork tapeworm requires a stool examination to detect eggs and gravid proglottids in the feces. Additionally, a complete blood count or CBC may reveal an elevated eosinophil count known as eosinophilia, as these cells are responsible for combating helminthic infections like Taenia Solium. Finally, in an adult patient presenting with sudden onset seizures, neuroimaging via magnetic resonance imaging or MRI or computed tomography CT scan of the brain will reveal multiple small cysts less than 1 centimeter in size, containing a central scolex which is the worm's head. This distinct radiographic appearance is known as the dot sign. Praziquantel is the first-line antiparasitic medication used to eradicate adult pork tapeworms residing in the human intestine. For cysticerci, however, albendazole is preferred due to its ability to cross the blood-brain barrier.